The next section of the endocannabinoid system module covers the various receptors, ligands, and enzymes that comprise the endocannabinoid system in the body. The endocannabinoid system is an endogenous system comprised of receptors, ligands, and enzymes. The endocannabinoid system, also known as the ECS, is not unique to humans. It is found within all animal species other than insects. The fact that the cannabis plant contains compounds which can act upon the ECS in humans is evidence of a common genetic ancestor for plants and animals. Some basic terminology that will be used throughout this module is defined here. The ECS is comprised of the largest receptor system in the human body, which has been broken down into two main types of receptors, the cannabinoid 1 and cannabinoid 2 receptors, known as CB1 and CB2. Both CB1 and CB2 receptors are part of the G-protein coupled receptor family. It is speculated that several other orphan receptors within the G-protein family, such as G-protein 55 and G-protein 18, may also be part of the endocannabinoid system receptor group. Cannabinoid receptors are located within every major bodily system, both central and peripheral, and their role is only just beginning to be understood as far as the therapeutic effects that can result from the manipulation of these receptors. Part of the reason cannabis can be used for such a wide variety of clinical indications is the massive proliferation of these receptors throughout the body. Cannabinoid receptor 1 is the most abundant receptor in the mammalian brain. It is encoded by the CNR1 gene, which consists of 472 amino acids in humans. Interestingly, several variations of the CNR1 gene have been associated with the development of cannabis dependence. The full-length CB1 receptor dominates in the brain and skeletal muscle, whereas a shorter allele of the CB1 receptor, known as CB1 receptor B, shows a higher expression level in the liver and pancreatic islet cells, where it is involved in metabolism. Previous studies have shown that in a system expressing the receptor endogenously, stimulation of the CB1 receptor leads to the activation of mitogen-activated protein kinase signaling pathways that are involved in the regulation of cell proliferation, cell cycle control, and cell death. This figure represents the major localization sites and associated functions of the CB1 receptors in the human body. The majority of the CB1 receptors in humans are found in the brain, where they are involved in various neurological activities. Although prominent in the substantia nigra and the periaqueductal gray matter of the brain, CB1 receptors are distributed in a limited fashion otherwise in the brainstem and not at all in the medullary respiratory centers, which is important in understanding why medications that interact with the endocannabinoid system and receptors do not cause the respiratory depression seen with opioids and other medications which do have significant medullary activity. CB1 receptors on the peripheral sites of the body participate in the regulation of local tissue functions. CB1 receptors modulate the mobility of the gastrointestinal tract, the secretion of gastric acids, fluids, neurotransmitters, and hormones, as well as the permeability of the intestinal epithelium. Therefore, CB1 receptors can control appetite not only from the hypothalamus and central nervous system, but also through the regulation of energy balance and food intake from the gastrointestinal tract as well. Another example of how cannabinoid receptors affect the body is a study in which rodents were given an animal model of osteoporosis. Half of the rodents had been genetically engineered to lack cannabinoid receptors. When a cannabinoid drug was given to the mice, the osteoporosis was mitigated in normal mice with intact cannabinoid receptors, but there was no change seen in the receptor knockout mice. This study further elucidated the potential role of cannabinoid receptors in regulating bone density and represents just one of the multitude areas of study into the role cannabinoid receptors play within the body. Cannabinoid 2 receptors are encoded by the gene CNR2, which consists of 360 amino acids in humans. CB2 receptors are primarily associated with cells derived from the bone marrow lineage and play a large role in immune system regulation and function. Although the expression of CB2 in the central and peripheral nervous systems is comparatively limited when compared to the CB1 receptor, it is undeniable that the CB2 receptor plays an active role in neurological activities such as nociception, drug addiction, and neuroinflammation. An interesting biological property of the CB2 receptor is its high inducibility, with CB2 mRNA levels often increasing as much as 100-fold following nerve injury or during inflammation. Endocannabinoids are the ligands of the endocannabinoid system. The first two endocannabinoids isolated were arachidonyl ethanolamide, or anandamide, also known as AEA, and 2-arachidonyl glycerol, known as 2-AG. Anandamide was named after the Sanskrit word for bliss, as it was found to bind to the same brain receptors as THC. 2-AG, the second endocannabinoid discovered, has activity at both CB1 and CB2 receptors in the body. These are only the two most well-known endocannabinoids, but there are in fact hundreds of variations produced by the body. While other neurotransmitters are synthesized and stored in vesicles, the endocannabinoids are unique in that they are produced on demand in response to a deep polarization-induced rise in intracellular calcium or the activation of various metabotropic receptors when and where they are needed, and then are promptly metabolized away. Endocannabinoids are produced on demand from arachidonic acid and interact with the cannabinoid receptors in the body to exert their effects. Both are fatty acid molecules which are highly lipophilic and insoluble in water, making them challenging to synthesize in a way that is readily absorbed by the human body. Anandamide was the first endocannabinoid discovered. It is primarily catalyzed from N-acyl phosphatidylethynolamine, or NAPE, by NAPE-specific phospholipase D. There are other routes of catalyzation not involving NAPE-PLD, but NAPE-PLD is by far the most common formation pathway of anandamide. 2-AG was discovered after it was determined that anandamide alone could not fully reproduce the effects of THC in the body. The basal level of 2-AG is approximately 1,000 times higher than anandamide in the brain. 2-AG is produced on demand from diacylglycerol, or DAG, by either DAG lipase alpha or DAG lipase beta. Most of the 2-AG mediating synaptic transmission in the brain is generated by DAG alpha. The receptor activity of endocannabinoids is still being fully determined. We know that anandamide is a partial agonist with high affinity for CB1 receptors, but has little to no CB2 activity. We also know that anandamide has additional activity at receptors known as capsaicin or vanilloid receptors, the TRPV1 receptors, which are best documented for their role in synaptic transmission and pain regulation. 2-AG is a full agonist at both CB1 and CB2 receptors. However, it has only moderate to low binding affinity for both. Much is still left to be learned about how the endocannabinoids interact with our bodies to exert their effects. After the endocannabinoids fulfill their role, they are quickly metabolized away. Anandamide is metabolized into arachidonic acid and ethanolamine through fatty acid amide hydrolase, or FAAH. 2-AG is metabolized by monoacylglycerol lipase, or MAGL, into arachidonic acid and glycerol. Anandamide can also be subjected to oxygenation by cyclooxygenase 2. A synapse is the space in which one nerve speaks to another through the release of neurotransmitters. In most cases, the neurotransmitter is released from the presynaptic neuron and acts on the postsynaptic neuron, causing either excitation or inhibition of that cell. Within the ECS, however, that process is reversed. The neurotransmitters anandamide and 2-AG are released from the postsynaptic neuron and work in reverse on the presynaptic neuron in a process known as retrograde signaling. Thinking about this clinically, when a signal is being transmitted, the ECS is what stops that signal when its purpose has been served, meaning it stops the inflammatory process or pain message that is being sent. Alternatively, the ECS can also stop the transmission of inhibitory signaling, allowing for the signaling to occur, which can play a role in the regulation of body temperature and stress. Once the activity of the endocannabinoid system as an inhibitory signaling system is understood, the ability of the ECS to be manipulated through the use of plant-based or synthetic cannabinoids makes much more sense, as does the intense interest in drug development targeted at the modification of this endogenous system. Now we put all of the pieces together that we've learned so far. This is a simplified scheme representing endocannabinoid retrograde signaling mediated synaptic transmission. In the diagram, thin arrows indicate enzymatic processes, thick arrows indicate translocation, and a blunted arrow indicates inhibition. The endocannabinoids are produced in the postsynaptic neuron upon neuronal activation. 2-AG is biosynthesized from DAG by DAG lipase alpha, and anandamide is synthesized from NAPE by NAPE-specific phospholipase D. As lipids, endocannabinoids readily cross the cell membrane and travel in a retrograde fashion to activate CB1 receptors located on the presynaptic terminals. Activated CB1 receptors will then inhibit neurotransmitter release through the suppression of calcium influx. 2-AG is also able to activate CB1 receptors located on astrocytes, which leads to the release of glutamate. Extra 2-AG in the synaptic cleft is taken up into the presynaptic terminal and degraded into arachidonic acid and glycerol by MAGL. Anandamide activates intracellular CB1 receptors as well as other non-cannabinoid receptor targets, such as the TRPV1 vanilloid receptor. Although endocannabinoid retrograde signaling is mainly mediated by 2-AG, anandamide can activate presynaptic CB1 receptors as well. FAAH is primarily found in postsynaptic terminals and is responsible for degrading anandamide to arachidonic acid and ethanolamine. Note that there are several alternative routes for the metabolism of endocannabinoids depending on the brain region and physiological conditions at play.